Okay, so now we're on to a general sense. Again, we talk about this as just basic somatosensation, right? So general, so scattered throughout the body. Vibration, pressure, light, touch, tickle, itch, temperature, pain, proprioception, kinesthesia are all movement, right? Are all spread in a variety of places found in on the surface and within the body. Some of these, okay, not tickle necessarily, itch inside the body, but other things are found within the body. We have two of these are free nerve endings, and this is temperature and pain. Merkel cells are vibrations. These are ones we found in the epidermis. Pacinian corpuscles are the deep pressure. Meissner's is light touch, or tactile corpuscles. There's a hair follicle plexus, really for movement, and then the Ruffinian or Ruffinian are the bulbous, and they are part of our stretch. So proprioception are mechanoreceptors. So again, movement or physical change. I know we talk about a mechanoreceptor. These are for body position. Length and tension of the skeletal muscles have a lot to do, right, tell our brain where our body parts are at any given time, right? It's not really a sensation per se, but it is important for a lot of our sensory perceptions to know, again, as we talked about balance, needing to know where we were positionally. So these are scattered throughout, when we talked about the vestibule and the semicircular canals in the last video, these were really, that was special sensory. This is really a general sense throughout. We often find these in the joints, or very close to the joints. So we have these things we call muscle spindles, and you can see why, all right? And they are near the tendons. And then we're going to have some Golgi tendon organs, which are actually going to be in the tendons. Okay, so here's the way this works, right? So wrapped around these individual muscle cells, right? Skeletal muscle fiber, which we know is a cell, right? And we would see that as the muscle fibers are stretched, right, impulses are initiated. So action potentials start and go to the spinal cord, and they cause contraction then of the muscle. So when muscle is stretched, it is automatically stimulated to contract. And this, you're going to find, it is a common theme for some of these mechanoreceptors. We'll look at this again in some of these, in other places in the body, not just in the joints. All right, the Golgi tendon organs are indeed found in the tendons, and they, what happens with them as tension increases, so pull or stretch, and they inhibit contraction. So they really are the opposite of these muscle spindles. And their job, though, is to protect the attachment of the muscle to the bone. So because if it's stretched too much, we can tear this. So they provide some moderation of the contraction by preventing it, right? And overall, the two of these can help us maintain posture, which is really not a contraction in either extensors or flexors or any of the opposites, agonists or antagonists. It's a right balance. So maintain posture, protect muscles from being pulled from the insertions by excess tension as well. So again, that's our job here when we talk about the Golgi tendon organs. And of course, in knee jerk or triceps reflexes, calcanean reflex, these are all showing this idea that, again, if you tap these sensors, they're mechanoreceptors, and that creates stretch. And so they can contract or not contract, again, depending on what the situation is. All right. So vision accessories. So again, we're just kind of giving you a little bit of this somatosensation. We're not going to dwell too much on it. And we're going to go into our last special sense, and that's vision. So lots of hardware, right, accessories associated to optimize our ability to see. And we have an opportunity to see a lot of this in lab, right? So eyebrows and muscles for movement and opening and closing, and blinking from the orbicularis oculi, right? So levator palpebra is for the eyelid itself. The conjunctiva is this membrane that really protects and is continuous with the eyelids. cornea that sits over the chambers of the eyes, and of course the actual conjunctiva that goes up to the cornea, doesn't cover the cornea. So eyebrows, again, perspiration and sunlight, protective, right, to optimize, again, our ability to see. Eyelids lubricate. There's some tarsal glands. You can see the openings to these if you look at your eyelids, upper or lower. Protective, that opens and closes through the orbicularis oculi and the levator. All right. Conjunctiva protects and lubricates. Again, this is when you have pink eye. This is conjunctivitis, excuse me, where there's inflammation of blood vessels in this membrane that's otherwise clear. Lacrimal apparatus, we'll see this on the models as well. This is part of our tearing, and it lubricates and protects and washes things away. And then the extrinsic eye muscles we'll look at on our models as well. And we're seeing pieces of them here. These all insert on the sclera, this fibrous part, and move the eyeball itself. All right? So here we're looking at these extrinsic muscles, again, all of them having an insertion on basically the sclera, which is a tough fiber that contains the whole eye, outer part of the eye. As we're looking at the eye from the front and anterior view, we have above, we have below, we have to the side, and to the middle. Right? So here we are looking at a right eye of someone. And that rectus, because they're all straight, so they really are appropriately named. And then we have an inferior oblique, because it runs to the side, and we are going to have a superior oblique that connects through this structure we call, its tendon comes through this trochlea. Trochlea means pulley. So here's a superior oblique inserting through this trochlea. So altogether four of these, four that are rectus muscles and two that are obliques. Which cranial nerves will control? Well, those are going to be the ocular motors, right? The trochlears and the abducens, right? So 3, 4, and 6 all have a lot to do with controlling eye movement, whereas 2 is the actual vision. All right, so here, as we're looking at this cartoon, the eye is a ball, right, packed in place with fat in those orbitals that we saw, and the orbit itself for the eye is created by about 7 bones, cranial bones and facial bones. All right, but the ball is fluid-filled, yeah, surprise, and the structure is very important for our ability to see. It's change in the structure length, really, of the eyeball that determines whether we have normal vision or we have myopic or myopic visions or farsighted vision, nearsighted or farsighted. So there are 3 layers that we call tunics here. Just don't use the word, we use tunic as opposed to strata for layers. We come up with a variety of terms, right? And this is basically an outer fibrous layer, a middle vascular layer, and then the inner nervous layer. So the outer fibrous layer is the only truly completely encircles and contains everything, okay? And it's the sclera and the cornea. Most of this outer layer is sclera. So the whites of your eyes that you see, that's part of the sclera, and it continues all the way to the back. And we'll see this in pig's eyes and on our eye models. The middle layer, vascular, has the blood vessels, because this layer is not vascularized. There's no blood vessels in this. If you had blood vessels in the cornea, which is what covers the front, cornea right here, covers the front part. It has to be transparent. If you had blood vessels, we'd have trouble seeing. So we had no blood vessels here, so the next layer in needs to be vascularized, and it's also colored, and it's colored to help absorb light and try to maintain optimizing the light directed to the nervous layer, the innermost layer. This isn't quite complete because of this, the pupil. So it's almost entirely encircles the eye in the middle layer again, but there's a space here known as the pupil. And the pupil is just an anatomical space. The pupil is surrounded by part of this which is called the iris. So in the anterior we have the iris, and then the ciliary body, which is made up of a process and a ciliary muscle. Beyond this, we talk about the choroid coat, right? So choroid coat, choroid meaning color, is really from about here on back, all right? And then the inner layer is the retina. The retina, of course, does not extend, is not a continuous circle here. It's where we have all our photoreceptors, our sensors for light, so there would be no point in having them up here, right? So these are in the back. And then there's going to be a variety of other terms we use. Now, of course, all to see, we have to start an action potential in the retina, and it must be transmitted to the optic nerve and back to the occipital lobe where we have the primary visual cortex. All right, so going through what we just said slide by slide, sclera and cornea are the fibrous layer, and the sclera is a firm white and protects, maintains shape, provides attachment, as we said, for those rectus and the obliques. The cornea is transparent and allows light in, because now we're directing light through specific pathway here to try to optimize, get the most of the light back to the photoreceptors. All right, the vascular layer, this middle layer, again, has blood vessels and is colored. So the posterior part is the choroid coat, and it's associated with the sclera. It's very thin with these melanin, these black. So on your models, they always show it as a kind of brown. On the cow's eye or pig eye, it's black. And it's going to help to absorb any stray light, so it doesn't reflect within the eye itself. Right? The ciliary body, which is anterior and is comprised of the ciliary process and muscle, and then is continuous with the iris. So the iris becomes part of this. So smooth muscle, known as the ciliary muscle, which helps to control the size of the pupil, right? And it's also attached to the iris is smooth muscle, which controls the pupil. The ciliary muscle is muscle that's actually controlling the shape of the lens. So there's these suspensory ligaments, right? So they're tiny. You're not going to see them on the cow's eye, and they don't really show up on the models either. But there's these tiny ligaments that attach this muscle to this structure of the lens. The lens is just a transparent proteins, but its shape has to be changed to help, again, direct light back here. So there's a structure called the anterior cavity that is created by these two layers, okay? And there's also a posterior cavity, but we're in the front here. So in front of the lens, any fluid-filled chamber in front of the lens is known as the anterior cavity. Any fluid-filled chamber behind the lens is the posterior cavity. The anterior cavity is then, shown here, divided into an anterior and a posterior chamber. All right. So again, the anterior cavity is between the cornea and the lens, and the iris is the separating point. So if you're in front of the iris, your anterior chamber. If you're behind the iris, your posterior chamber. These are filled, so the entire anterior chamber is filled with aqueous humor, water-like humor, maintains pressure, helps to refract or bend light, and also provides nutrients to the cells here. All right. This fluid is produced by the ciliary body, and it comes from blood, because remember, the ciliary body is part of a vascular layer, right, and circulates through and then is reabsorbed. All right. People with glaucoma, there is a problem with either producing too much fluid or not draining the fluid away, and most likely it's going to be the drainage. So it doesn't drain fast enough, so fluid builds up in these chambers, and that's why we test for glaucoma by blowing air into your eye and seeing how much pressure is reflected from the eye. So glaucoma can result in blindness because this excess pressure in the anterior cavity can cause neural cells to die. So we don't want that. All right, the nervous layer, and I should say the posterior cavity, is going to be filled with vitreous humor, as I show you here. All right, so the nervous layer is the retina collectively, and there's several layers to the retina. So there is a pigmented layer, and this is continuous with the choroid. So here's this pigmented epithelium that's going to be continuous with the choroid coat, all right? And it is pigmented to, again, prevent any light that passes and doesn't excite these rods and cones. We want it absorbed, okay? So you'll see, here's our pigmented cartoon. Rods and cones, rods outnumber the cones. Rods are part of the your black and white, so not color, and cones give us, this color, ability to perceive color. So we have many more rods than we do cones. They don't require as much light to excite them either, so that's why when there's not much light out, things do not appear very colorful, because your cones require more light to start action potentials.